Okay. Welcome to Ung Botaniker Tromsø's Christmas celebration. Sending live for the first time, very exciting. And it's been a really odd year, but also a good one. So we had several plant trips during the year. Also, even with Corona, we've been able to have some plant activities. And tonight, we're lucky to have Isa here to tell us more about machine learning in biology. So excited for this. Welcome to Isa.
Thank you. Thanks for having me. A small Corona-friendly crowd and a live audience. That's awesome. Yes, as I said before, if you've just joined, if you have any difficulties hearing or seeing anything, please just let us know in the live chat. And if you have any questions also here in the audience, you just raise your hands or post the questions, and you can do that during the talk, but there will also be time for questions in the end. So let's get straight to it. PollinatorWatch, combining biology, machine learning, and citizen science. I'll try to see if I can stay in picture. Just starting with a little bit about me. My name is Isa, and my background for being here is that in 2015, I started a bachelor's in cognitive science, which might not have anything to do with biology. But then in 2018, I did an exchange here at UiT, my first time in Tromsø, so almost exactly 3 years ago. And then I went home again and I started a master's in cognitive science at Aarhus University. And then I sneaked my way back up here this semester for an internship at UiT in collaboration with Aarhus University, the Department of Bioscience at Aarhus University. Basically, I come back here every chance I get because it makes my face look all happy and windswept, and there's mountains and snow. So yeah, I try to come back as often as I can. Let's get right to the first part of the talk, which is about biology. I've also called it preaching to the choir, or the part that is really not my field of expertise, but rather yours. But please bear with me. First, we have to define what a pollinator is. So, as you probably know, plants pollinate by transferring pollen from female to male species, and then they produce seeds. And so a pollinator is basically any animal that transfers pollen grains from one flower to another. This means that not only bees and flies, but also bats, birds, and even some mammals can be considered pollinators. And because of the way pollen is transferred with these pollinators, many plants are dependent on insect pollination for their survival. Because without them, they're not going to be able to produce seeds that's going to produce the new plants. So that is all good, but there is a problem. So here we have a quote from Toge Thomas Høje, who's my supervisor on this project, a senior researcher at the Institute of Bioscience from Aarhus University, who says that this is really hard for me to read. We see on a global scale that populations of insects are broadly in decline, and we have a limited understanding of why that is. So one thing we do know is that 1/3 of all bee and butterfly populations are declining. 10% of all bee and butterfly populations are endangered, so they are at the risk of disappearing completely from our ecosystems. And just this morning, I found this press release from the Fish and Wildlife Services, who suggests putting the monarch butterfly on the endangered species list, but basically they can't because they're too busy protecting other species that are going extinct. So this is a big problem, but why is it a problem for us? Well, first of all, 75% of all human food crops worldwide depend, at least in some part, on pollinators. So without them, we're not going to be able to feed the world's population, basically. And it's a good thing we're not paying these pollinators for their work, because it's estimated that roughly $3 trillion a year's work is done by pollinators, and that's kind of a byproduct of their just going around. So we're really lucky that they're doing this work and we don't have to pay for it. So obviously we want to find out what's going on here. Why are pollinators disappearing? And the way you do that is you do some biological research. And you all know that traditional methods from ecological research often include fieldwork, manual identification of species, and in my own limited experience, a lot of counting of things. And obviously fieldwork is awesome and fun, but it is also time consuming and it has some spatial and temporal limitations. You can only really be in one place at once, and there's so much going on in an ecological system that if you take just 1 square meter at traditional biological methods, as far as I know, and look at that, there's a lot of things going on in here. So it's really labor intensive to do this kind of research. So what if we could optimize this in some way? That's what Toge started to think about when he came up with this research question or this research project. So what he said was that an automatic system for identification of insect species would be a game changer for this kind of diversity studies. So what's meant by automatic? Well, his idea was that you could use time-lapse images, set up cameras at various areas around the Arctic, and then take pictures continuously of clusters of flowers to watch the plant and pollinator interaction going on. That way you increase the temporal scale because you're taking images continually, and also the spatial scale because you can set up more cameras. You can basically be in more than one place at once. Which brings us to the second part of my presentation, which is about machine learning. It's a little closer to my field of expertise, but still you should remain very skeptical about everything I say. So first part of this project was to set up the time-lapse images. This is a little GIF I created of a day in the life of a Dryas integrifolia, which is the Latin name of this flower that grows in clusters in various Arctic places. And this is just one day, the 8th of June 2017, in Narsarsuaq in southeastern Greenland. And as you can see, the sun never really sets during this time lapse because in the Arctic the sun is up for 3 months a year, so you have plenty of opportunity to capture some data. And this is only one image per hour, but the real cameras obviously took way more images than this, several per minute. So you end up with quite a lot of data when you do this in several locations in several countries for several years. So what do we do with all these images? We feed them into a convolutional neural network, of course. That's the modern solution to everything. Convolutional neural network is just a fancy way of saying that it's a neural network that's optimized for two-dimensional matrices, such as images. And you might know this very classic Exposity comic. It's probably going to be a little hard to see on the feed. But one guy walks up to another guy and says, "This is your machine learning system? Yep, you pour the data into this big pile of linear algebra and collect the answers on the other side. What if the answers are wrong? You just stir the pile until it starts looking right." And that is a little bit meant to be funny, and it's also a little bit meant to be the truth. Because we don't really know what's going on in this big pile of linear algebra. So all we can basically do is to feed the network good data and hope that something good comes out in the other end, something that we can use. So what Hjalte Mann did, who is the PhD working on the machine learning part of this project, is that he created a very cool convolutional neural network that could take all of these time-lapse images and cut them into crops containing only the flowers, so cutting away basically all the ground around the flowers. And what we ended up with was images that looked kind of like this. and we've now created even more data. So for each of these many images, we now have divided it up into upwards of 20 flower crops. So we've basically solved one problem, the temporal and spatial limitations of ecological research, by creating a new one, which is we have so much data, we have no idea what to do with it. Which leads us to the third part of this talk, which is about citizen science. And now we're getting kind of close to something that I know a little bit about. So first off, what is citizen science? I'll start you off with a quote again by Resnick et al. So citizen science is a range of collaborative activities between professional scientists and engaged lay people or citizens in the conduct of research. So citizen science is basically having non-scientists or volunteers collect or transcribe or categorize or analyze your data for you. It's a little wind going on here. Citizen science has a great potential for knowledge sharing. You can involve some people in some research that wouldn't otherwise have known anything about it. It can become kind of like a hobby for some people, that they can do science instead of watching Netflix maybe. And it can also be said to democratize research, because it's not just for the elite anymore, but anyone can actually participate and get something out of it. So the way we chose to implement citizen science into our project was with a platform called Zooniverse, which is the world's largest online citizen science portal. They have launched more than 200 citizen science projects. They have more than 160 peer-reviewed papers published with data from volunteers, and more than 1.6 million users on the site. So that's a really big resource to tap into if you can get your project up on this site. So that's what I've been working on since August, and that was a really slow process because it has to go kind of through different improvement steps. And I'll say, if I had done this talk just a week ago or 2 weeks ago, it would have been very different. But as it happens, we launched our Zooniverse project yesterday evening. And it went really great. We went live at roughly 5 p.m. And exactly 4 hours later, I tweeted out this: we've already had more than 30,000 unique classifications from users on the Zooniverse. So that was just fantastic. And just to give you a little preview of what the project looks like, maybe I can't do that. Can you see this? Yeah. Oh. There we go. No? Oh. I'm out. Okay. Oh. Okay. Technology. Okay. Here we go. This is PollinatorWatch, and just scrolling down here, you can see that yesterday evening we ran out of data after 4 hours, so I uploaded 40,000 more subjects, and we've now had 75,000 classifications since last evening, which means that we have just rounded 100,000 unique classifications in less than 24 hours. So that's really great. It looks like this. When you come in, you can learn more about the project, or you can get started classifying images. So when you go in, you get a little prop that looks like this, and then you can say, does this contain a pollinator? Yes or no. And then you just keep going until you find one. And then you keep going. So that's kind of what it's about, just to give you a little taste. And now I have to navigate back here. Here we are. Okay, so what we want to do with PollinatorWatch is that it kind of has 3 phases, and we just got started with the first one, which is to find roughly 2,000 pollinator crops that we could use to train a neural network to automatically find the crops. So we're basically using volunteers to automate their own job. And then in the next phase, we want the volunteers to then kind of check this new network to see if it did what it's supposed to do. So the images that the new network said were pollinators, were they actually pollinators, or were they just false positives? So maybe a stone or something else, or maybe even a flower without a pollinator. But ultimately, the goal of PollinatorWatch is not to be able to locate pollinators in crops of Trifolia from southeastern Greenland in 2017, because that's way too specific. The goal of the project is, of course, that we want to be able to locate pollinators everywhere in all kinds of environments, and maybe even in live images or video. So the future for PollinatorWatch could maybe be to have volunteers distinguish between different kinds of pollinators, or it could be that we have new kinds of flowers or undergrowth that we look at. It's really hard to tell right now. It very much depends on what comes out of these first phases. But it's going to be really cool. I'm pretty sure about that now after we've launched. I wasn't so optimistic before, but now I'm pretty optimistic. Okay, so which brings me to the last part. What does any of this have to do with cognitive science? I'll start you off with a quote from my supervisor, not Toge, but my AU supervisor, my cognitive science supervisor, who said, make sure that you can argue that this actually has something to do with cognitive science, and it's not just you going back to Tromsø to play with your biology friends. So I guess I'll have to try and make that argument now. Okay, so PollinatorWatch is kind of like a game, you could say. Our goal is to keep people engaged and to play for so long that they do a lot of annotations and we get a lot of data out of it, and at the same time they have fun and feel like they are getting something out of it. So one thing we wanted to investigate in the beta testing stages of this project was, can changing the workflow make the annotation process more engaging for the volunteers? So the workflow we saw before looked a little different from this one. This was our first workflow, and you can see that it has 3 different options. You could either choose if something was a pollinator, a flower in bloom without a pollinator, or a flower that wasn't blooming, or just simply a false positive. And after testing that out for a while, we realized it was a bit clumsy and it wasn't so easy. And we wanted to test out what would happen if we changed to the binary workflow. So would this make it more fun and quicker to go through the annotations? The binary workflow also allowed us to create what has basically become Tinder for pollinators. So if you go into the Zooniverse app, you can just swipe now, because we only have 2 options. So is it a pollinator? Yes or no. So we wanted to see if this made a difference. These are the initial results, and this is probably going to be hard to see on the live feed, but we have a blue graph, which is the original workflow, and a red one, which is a little shifted to the right, which is the binary workflow. And on the x-axis, we have the number of classifications per user. And what you can see, the lines are the means. So when we went from the original to the binary workflow, we saw that the mean number of classifications increased from 30.5 to 180.3. That's a 300% increase. So that was great. That was really something that worked, and keeping people engaged. Another question you could ask is, how many pollinators do we need to keep people engaged? So we know that in the original data, there is not a lot of pollinators compared to flowers without pollinators. But if we just feed people the raw data, you have to swipe through like thousands of images before you see one pollinator, and people are not going to play that game for long. So what we did is we manufactured some data sets that contained more pollinators, so images that we already knew were pollinators mixed with some images that we didn't know. And we started off with just 4%, just to try something. So that means that every time people swipe through 25 images, on average, they should see a pollinator. One thing here that's important to mention is that we obviously want this number to be as low as possible, because people looking at images that we already know contain pollinators is basically a waste of time for them and us. Of course, we could use it to quality test our volunteers to see if they actually hit the right pollinators, but once we've done that, it's kind of just wasted energy. So we changed it down to 2%, and we again had an increase in the number of classifications. It went up by 180% roughly. I will say, though, of this number that it could be caused by other things, such as we were a bit further in the beta testing stage, so some of the volunteers that didn't like the game might have just dropped it and not played it anymore, which would draw the average up. So we have to test this a little bit more. Yes?
Was this an increase from the binary or from the base level?
This was an increase from the binary.
Okay.
Yeah. Yeah, so but we stayed with the 2% for the launch, and we've already had one volunteer write to us that it's boring when you don't see a pollinator often enough. So we'll have to try some different percentages to see what works best. Yep. I just have one final slide for you, which is about what you can do to befriend a pollinator. I just wanted to end with this because I think it's a really important issue. And the first thing that you can do is really easy. It is to do nothing. Basically, the more patches of unmowed grass, the more leaves and old shrubs and branches you can leave around for the pollinators, the better. It's less work for you, and it's good for the pollinators. So that's a good one. The second one is to plant wisely. Young botanists know this and are probably really good at it. But choose plants that are native to your environment. Remember, a plant is only a weed as long as it is unwanted. It's just a name we've given some plants that come up by themselves and that we don't want in our garden. But actually just allowing things to grow wild can turn into something really beautiful, as you'll see an example of here on the right. This is from my own little city garden at home in Aarhus, where some bronze fennel has just been allowed to stand and go into bloom. And the pollinators love that. Finally, you can PollinatorWatch with us, sign up to Suniverse and Advanced Science while lying on your couch, eating potato chips in your pajamas during your Christmas break. And we really hope that you will do that. Thank you so much for listening to me. You can get in touch with us at Suniverse or Twitter on PollinatorWatch. We are Tove Høje, Hjeldemann, Rebecca Veen, and me, Iselø. And now, if you have any questions, feel free to